Hey, what is going on, my peoples? It is January 22nd, 2022. And yesterday, for those of you who are paying attention, you might have noticed that I missed a day of recording. Yes, it was really busy and I had some really weird stuff happen with my vehicle. Forgive me, I'm drinking coffee with whipped cream. It's really good, but don't overdo it. I had some stuff going on with my vehicle, and I did not get a chance because of work in the day to record this video that I love recording. And yeah, so I'm here now to talk about to go back to get back in the swing of things. And I'll just say that just to refresh you and everyone else of where I was. Okay, I was talking about the lymphatic system. Okay, yeah, and that was kind of wrapping up my initial foray into the immune system, like the encompassing the high school biology level, right? So I went from the immune system, the basics, starting with the innate immune system, the complement system, the adaptive immune system, the actors, which are the leukocytes and the lymphocytes in those systems, to the way complement protein behaves kind of instantaneously and how it's an ancient system, and how the innate immune system needs help from the adaptive immune system to be more specifically targeting pathogenic invasions, and all the way to blood types and other parts of the immune, and basically immunosuppression, how immunosuppression works. I've just went through the whole gamut of things, and even up to cancer, and not as much as I need to, because I'm gonna go revisit that stuff later, because the next go around, which will be coming soon after I go through my current series, is going to be going a level deeper into the chemistry of the immune system and really tying a lot of the pieces of the immune system together in the intracellular level, which is very important because on the high school biology level, most people talk about the extracellular and cell-mediated immunity and humoral immunity and antibody production through B cells and activation by helper T cells, etc. But then, inside the cell, when the signaling occurs, when a cell receives these interleukin signals and just needs to react, what actually happens? That's where I want to get to, right? So, but we'll get to that soon. We need to actually take a tour, or at least I think I need to take a tour into the blood, because the immune system is great. Well, the root of the immune system, the leukocytes, right, and lymphocytes, those really need, those have a medium of transport, right, and that's a very important, the circulatory system is a very important part of your body. It's a critical part of your body, so we should understand both, because they both work in concert. And of course, all parts of your body essentially work in concert, but these two, I feel, are the closest together of working in concert. And of course with your nervous system, which drives everything. But anyway, so we're going to talk about hemoglobin today. All right. What is hemoglobin? Hemoglobin is one of those molecules that even rappers know about, right? And they rhyme stuff with Jeffrey Tubin and hemoglobin. I don't know, but it's probably the most well-known, and up there in terms of the most important molecule complex, molecular protein complex that's needed by your body to and your red blood cells specifically, and your red blood cells to do their thing. We also need to talk about myoglobin, right? And myoglobin is not as well known, but it essentially functions in a similar manner because it has the same mechanism of oxygen binding, fundamental mechanism. So we'll start with hemoglobin, and that hemoglobin is this molecule which resides in your red blood cells. So if you take a look at this right here, this is kind of roughly, in a very diagrammatic way, where hemoglobin lives, right? And this is basically what happens, right? The oxygen molecules are traversing through your bloodstream, and then the red blood cell with hemoglobin embedded comes by, and by the way, each red blood cell has many hemoglobin molecules, right? Like there's not one, or there's not just this one sitting in the center, right? They're peppered, and I don't know what the number is, but I think it's in the hundreds of thousands per red blood cell. But anyway, what happens is red blood cells are erythrocytes, right? They have these heme groups, which the heme group itself is this structure from the top. So there's two things to consider, right? The heme group is actually this molecular structure here, which are all these rings, which are binding central nitrogens, which bind a central iron. And of course, the iron is the important part, the most prevalent actor in this molecular complex here. But this is not, we can't only consider this two-dimensional iron here. What this iron does, this iron essentially binds the O2, right? There's obviously oxygens, in the periphery of this atom in different organic groups, but the central iron is what is responsible for the transportation of oxygen. So essentially, it's kind of like an interesting trigger system that works. And obviously, I know this is, for some people very easy, but we will kind of like see what's happening here. So the molecular diagrams can get pretty complicated, right? But trying to find a good one that kind of describes what's happening. Okay, yeah, this is a pretty good one, right? So you have your heme group in a three-dimensional plane with this iron in the center. So you notice this iron is sort of, if you notice carefully, these bonds are bent downwards, because connected to this iron you have this histidine amino acid, right? And this histidine amino acid has a name. It's called the proximal histidine. Okay, we'll see why it's classified that way, but it is essentially, if you take this plane, it's on the bottom of this plane, and due to the quantum mechanics and bond geometry that is caused by these forces, bond forces pulling on this iron, you have this distortion, right? Which I'm sure the actual angle of these bonds have been measured for an iron through this central iron through spectroscopy, but for now we'll just say they're bent, right? So what happens is then when the actual oxygen binding occurs, the O2 dioxygen binds to this iron, right? And then this iron then stabilizes, and the plane of the iron flattens out. Okay, so that has a bunch of consequences, but essentially that's in the bound state for oxygen. And how does this oxygen stay there bound to this iron? Well, this oxygen has a resonance structure between dioxygen and superoxide. So because if the oxygen wasn't stabilized somehow, then the oxygen would leave, because there's an unstable attraction between the iron and the oxygen molecule there. So how does this bond stabilization occur? Okay, so what happens is essentially another histidine called the distal histidine comes by, and comes by, it doesn't just come by, it's chemically bound and probably enzymatically driven to be bound to the dioxygen. And the distal histidine that's bound to the dioxygen, it's not shown here, but trust me, that's what happens. It then stabilizes this oxygen and turns it in, it gives it a certain resonance structure, which allows the whole molecule to stabilize, and essentially the hemoglobin to be, or the heme group to be triggered into place. So let's see what happens. Then the hemoglobin itself, now hemoglobin is the blood transportation molecule of the system. There's actually, if you call these heme groups, these circular things here, these were that molecular structure here that we just looked at. Each of these you can imagine is this ring here, right? and each of these has a central iron in the Fe2 state, ionic state. So it's ferrous, and then when the when the dioxygen binds to the iron, and with the help of the distal histidine, it becomes in the ferric state, which is Fe3 plus. Okay, so Fe2 plus goes to Fe3 plus. Anyway, so there's 4 oxygen binding sites on a hemoglobin, right? So you have these, and the hemoglobin, the rest of the molecule is this long amino acid protein, single chain amino acid protein that's folded in all these complex conformations, and then attached. These are attached to each other, right? And the chemistry of this attachment has been studied, obviously. So, and the alpha, beta, alpha 1, alpha 2, beta 1, beta 2 chains, they're similar to each other, and they all bind hemoglobin. And so, and myoglobin itself, myoglobin is actually a much simpler molecule. It's a single.
So I've got my essay.
Wow, it's a single polypeptide chain, which it's a single protein amino acid chain, which has a single heme group on it. Okay, so here's the question: if you have hemoglobin to transport oxygen throughout your system, and you have myoglobin also, what do you need both? Okay, essentially the difference is where the myoglobin and hemoglobin actually act in their functions. Okay, so let's go to the oxygen binding curves of myoglobin and hemoglobin. So basically, this is the oxygen. This is the rate at which oxygen is bound to the percentage of oxygen binding of myoglobin versus hemoglobin, and at which pressure, because the variable that affects what the binding affinity is, or rather the oxygen saturation is of myoglobin versus hemoglobin, is this pressure of how much oxygen is in the environment, right? So myoglobin, you see, binds oxygen with a much higher affinity when the pressure is much lower, right? And if you think of this as an association curve, oxygen association curve, in the reverse, you have the dissociation curve, which is the same. So dissociation occurs at a much lower pressure for oxygen. So for myoglobin. So what that means essentially, this myoglobin is good at holding on to oxygen, whereas hemoglobin actually the oxygen pressure needs to be much higher for hemoglobin to bind to oxygen, right? So essentially you have to be, as opposed to a very tiny amount, you have to be about 50% before you start to see close to 80% saturation in the sigmoidal curve. That's what this S shape is, right? So this hemoglobin then also in the reverse direction disassociates faster from oxygen, from O2. Now, why is this the situation? Hemoglobin has more heme groups, right? Myoglobin has 1, hemoglobin has 4. So that essentially boils down to 2 considerations: the function of hemoglobin and myoglobin versus myoglobin, and the structure of hemoglobin versus myoglobin, and the binding mechanism. So hemoglobin has 4 heme groups, that's true. But the binding affinity is affected by the cooperative nature of how these oxygen are bound. So it's quite complicated, but essentially what happens is hemoglobin, when a little bit of oxygen is present, it doesn't bind that oxygen too vigorously. But the more oxygen that's present, the quickly that hemoglobin binds. And this is called cooperative binding. Okay, so hemoglobin cooperatively binds to oxygen because it uses the help of the heme groups that are already bound to increase the binding affinity of the unbound heme groups. So that's a mouthful, but it means that the more oxygen pressure there is, the more binding that occurs, the more more likely the heme groups that haven't been interacting with oxygen yet are to bind to the hemoglobin. So why is this needed versus myoglobin? So hemoglobin is the as the oxygen transportation molecule complex of the blood, right? And myoglobin actually lives in your muscle cells, and that's why it has the prefix myo for myocyte, right? So myoglobin is needed because oxygen storage in your muscles is needed for when activity occurs and the oxygen is needed right away for activation. Because hemoglobin needs to take up oxygen and then needs to easily drop off oxygen. Myoglobin needs to hold on to oxygen. So myoglobin is a storage molecule, whereas hemoglobin is a transportation molecule. Hemoglobin disassociating like this and associating like this is a consequence of where the hemoglobin picks up the oxygen in the first place, and that's your lungs, okay? So hemoglobin is, remember, only going to pick up oxygen when oxygen is available, when the oxygen pressure is high enough. And where is the oxygen pressure highest in your body? It's going to be in your lungs, where you're obtaining that oxygen. At that point, hemoglobin can pick up the oxygen really easily, but the disassociation, it needs to get rid of the oxygen fast as well, right? Compared to like myoglobin. Myoglobin really needs to hold on to the oxygen until it can't hold on anymore. Hemoglobin needs to get rid of the oxygen as quickly as possible. So wherever the hemoglobin ends up, right, it starts to disassociate oxygen, and the more that is disassociated from it, as the oxygen pressure decreases, the faster the oxygen leaves. So it's basically a trigger for the oxygen to spill off the hemoglobin and go to where it's needed, or be transported to where it's needed and what tissues it's needed at in the body. So that's basically the difference there, right? So muscle cells need quick activation and uptake of oxygen and need to hold on to oxygen for as long as possible during their activity cycle. That's why this myoglobin releases it. And this myoglobin does it all on its own. It doesn't have the help of already triggered myoglobins to do so. Therefore, it's called uncooperative binding, right? Not cooperating with anything else. So that's quite a bit for this one lecture. I've gone almost 20 minutes. But yeah, let's move on this to another. We're going to go deep into the blood system, so to speak, for a while, so we can fully understand what's going on there. And then we'll tie everything together and go back to the immune system and the nervous system. Okay. Thank you for listening. I'll see you later.